So, building vocabulary for this section 13.1. Transformation is a process in which one strain of bacteria is changed by a gene or genes from another strain of bacteria. A bacteriophage is a type of virus that infects bacteria. If you break the term bacteriophage down into two parts, bacterio and phage, it may help you understand it better. From the definition, you can see that the phage is a type of virus. With bacterio at the beginning of it, you can see that the relationship between the two parts of the word. Through experimentation and watching the process of transformation in bacteria, scientists learned that DNA stores and transmits genetic information from one generation to the next. About a century ago, scientists who wanted to understand genetics better began experimenting to learn the chemical nature of genes. In 1928, the British scientist Frederick Griffith was investigating how certain types of bacteria produce pneumonia, a serious lung disease. Griffith had isolated two very similar types of bacteria from mice. Both types grew well in culture plates in Griffith's lab, but only one of them caused pneumonia. The disease-causing bacteria, the S type, grew into smooth-edged colonies on culture plates, whereas the harmless bacteria, the R type, produced colonies with rough edges. The difference in appearance made the two types easily to tell apart. When Griffith injected mice with disease-causing bacteria, they developed pneumonia, while those injected with harmless bacteria remained healthy. An injection combined heat-killed, disease-causing bacteria and harmless bacteria still made the test mice sick. Griffith identified that a chemical factor turned dead and harmless bacteria into disease-causing bacteria through a process called transformation. He determined that the disease-causing ability was transferred to the bacteria's offspring. Thus, transformation was caused by a gene. In 1944, Oswald Avery and a team of scientists tried to repeat Griffith's experiments to identify the molecule in the heat-killed bacteria that caused the transformation. They first removed molecules from heat-killed bacteria and used enzymes that destroyed their proteins, lipids, carbohydrates, and RNA. Despite this, transformation still occurred. A second experiment, where enzymes were used to destroy DNA, proved that when this had happened, transformation did not occur. Their experiment proved that DNA must be responsible for the process of transformation. Experiments with bacterial viruses demonstrated that DNA and not the cell's protein coat carry genetics material. A bacteriophage is a virus that infects bacteria. One way bacteriophages infect bacteria is by inserting genetic information into a cell and reproducing until the bacteria burst. To determine which part of the virus entered the bacterium, Hershey and Chase grew viruses with radioactive isotopes. These identified which molecules entered the bacteria, showing that DNA, not the protein coat, held the genetic information. DNA stores and copies genetic information and then transmits it to the offspring. Through DNA, genes are expressed and cells develop with specific characteristics. DNA's primary job is to store genetic information. It is the heredity molecule, and it controls cell development. All information for a single cell to develop into a complex organism is stored in DNA. DNA's second job is to copy all of the genetic information exactly. DNA's third job is to express the genetic information into other cells so they develop into exactly what they are coded to be.